All right. Welcome back, everyone. Hope you got a nice brain break and tasty food. I forgot to mention, I'm sure you've noticed by now that we have a coffee station over here on the right, so help yourself at our next break. So I will go ahead and leave it to Melissa Thalen, our research chair for the foundation, to introduce our next speaker.
Perfect. Thanks, Carly. And I wanted to kind of introduce Kyle, our scientific officer, and our journey in getting to having a scientific officer as the NR2F1 Foundation to get us started. And this is a little timeline I created that kind of gives you a snapshot of what's been happening behind the scenes. I think we've shown some of this on the Facebook page, but just to help you have where we've been going. So in 2020, we ended up applying for the Zuckerberg Initiative grant, which was the first time that we really had to sit down and formalize what is the right path of research for BBSOAS. And it was enlightening to kind of realize there was so much more we did not know than what we know. And kind of in parallel to that, we also went to the Disorder Film Festival that I mentioned earlier, and we were in the room with some other rare disorders, NGLE1, SYNGAP, USP7, and these people were just much further down the path, identifying the right direction, and they had cell models and mouse models, and I was just in awe. And so I thought, wow, how cool would it be if we were in a room with these types of people more often? And in parallel, Carly learned about the opportunity for COMBINEDBrain, and that is something Terry Jo founded and will speak on this afternoon, but it's really given us that opportunity to learn from all these other disorders who are further down the path and avoid some of the missteps they have made in what our ultimate goal is for any neurodevelopmental disorder and getting to a clinical trial, actually establishing treatments that will have an impact. And so we were, still identifying the right research roadmap for us and decided there's probably some great intellectual capital out there with university students who are just also learning about neuroscience and might be willing to step up and help us. And so we created Project NR2F1, reached out to, I think, the top 25 neuroscience programs across the US, and in the end, received 5 proposals for different paths we might use for studying BBSOAS further, things like using gene therapy and targeting the eye, things like using CRISPR to do some screen drug tests, and then also the idea of looking at a mouse model across various points in time and really identifying how NR2F1 was impacting the development of the brain. And that was ultimately the winner. Dr. Shaw had some influence in speaking with that group, so really good direction there. And as we identified, the Project NR2F1 path, and we're catching up with Terry Jo just on a regular check-in of the progress we were making, she mentioned having the Rent a Scientific Officer program as part of COMBINEDBrain. And ultimately, we thought this is an amazing opportunity. We all have full-time jobs and are, juggling a million different things. None of us have the time or really the background and education to be insightful and as thoughtful about this as we'd like to be. So that's where Kyle came into play. He works very closely. We meet twice monthly and just talk about, he's gone through all the research now. He's really taking that Project NR2F1 roadmap to a much more strategic level. I will let him share more of his credentials, but ultimately, we've already received our first grant with his help, and we have a collaborative now where we've identified some potential treatment targets. So we're really moving in much faster than I would have ever believed possible, all the right directions. So with that, Kyle, I will let you share a little bit more about what you're working on.
Okay. All right. Thank you, Melissa. Well, I will start introducing myself then. I don't need slides for that. So, yes, I'm going to be, there it is. talking about developing a research roadmap for BBSOAS. So let's make sure this works. Perfect. So yes, thank you, Melissa, for the introduction, and thank you to the NR2F1 Foundation for inviting me to speak. So I am fairly new to BBSOAS in comparison to other speakers we've had today. I've been working with the NR2F1 Foundation since July of last year. And so I am the scientific officer for the NR2F1 Foundation. That is sort of my part-time gig. I am a postdoctoral fellow at COMBINEDBrain. And as Melissa mentioned, COMBINEDBrain has a rent a neuroscientist program that the NR2F1 Foundation had taken advantage of. So I put up there our mission statement for COMBINEDBrain. So generally, basically, we are a nonprofit. We are a group of patient advocacy groups, and we work with those groups, clinicians, researchers, and pharmaceutical firms. And our main goal is to speed up the time from for rare disorders being identified and then moving that to clinical trials or pursuing treatment options as fast as possible. So I was asked by Melissa to basically develop a strategic plan, a research plan for NR2F1. And so what that really entails is that, let's see. So as a scientific officer, basically my job is to understand all the research, so go through everything that's published so far, so know the research field, and be able to advise the foundation on a scientific perspective as to what to focus on, what to pursue, what to allocate funds to. And so basically what a scientific officer does is know the field and be able to advise what to do next. So you might also describe it as a consultant or a scientific consultant. So developing this research roadmap or scientific strategic plan is one of my part of the job of a scientific officer. So let's see. Okay, so my assignment was to assess the landscape, so go through the literature, meaning published case studies, clinical reviews, anything that's available on PubMed, which is the general database for biological research. So go through what is known about BBSOAS, including cell models, mouse models, and the idea is to assess, take in everything that we know, everything that's known scientifically, assess everything that is available in terms of resources to the NR2F1 Foundation, and then say, okay, well, what is most critical that is missing, and then how do we fill in those gaps? And by doing this, we are kind of following in the path of other previous rare disorders and what they've done right and what was successful for them. So a lot of this, or most of this, is what you would call translational research. So you may have heard of this term bench to bedside, and what that basically means is being able to connect what is done in a research lab by basic scientists, so what they are doing and what is possible, and connecting that to what is done in practice by doctors, what is done in actual practice. So the goal is to develop treatment strategies as our long-term goal, and integrate everything that is known, find the fastest pathway to treatment. So just a quick review of the literature, 2018 is significant because that is the last time that the NR2F Foundation met for a conference. And at that time, there were just 4 publications describing BBSOAS in PubMed, which is the major database. As of 2021, or at the end of last year, for a project I was going through and seeing everyone that works that has an interest in BBSOAS, including different scientists, research groups, and doctors. And I had counted 14 at that time. That is not a, so I would assume that by this point, that number is either a bit higher. But the major point is that 2022, so just 4 years later, we are up to 25 different publications. That is up from 4. So the main point is that we are growing. The research field is growing at a faster rate, and that is quite exciting. So in addition to the 25 publications, there are other publications about NR2F1, which had been studied for a while before BBSOAS was identified. And so there are other publications that have direct relevance to BBSOAS that doesn't mention it specifically. So I'm showing this figure from a publication that came out just 2 months ago, and it was the largest and still is the largest description of BBSOAS patients at the time. And so the purpose of this figure is of showing you this figure is twofold. One is to show just the enormous amount of data that is being collected, and this is just a tiny fraction. And then if you were to interpret this figure, so on the top, you can't read these, but on the top, going across horizontally are different patients, and so they had looked at 84 different BBSOAS patients. And then on the right side, going down are different symptoms or different phenotypes, and they took the most common ones. So just looking at the most common ones, so a yellow square means that they don't have the symptom or the phenotype, and the red square means that they do. And so if you're just to compare one column to the next, you could see that none of these patients are identical. So there's quite a bit of variability, which previous speakers today have spoken about. So there's quite a wide range of variability. There is quite a wide spectrum. So part of my task is using this template of questions that we developed. And so basically we ask a series of objective questions of what is known, what is available to the NR2F1 Foundation. And we use this as a rubric to kind of guide our recommendations to the foundation and to steer BBSOAS research. And so I've broken this down to 6 parts. So I have this here, and so I put this sideways so you don't have to read all this. This is just a sampling of some of the questions that we have describing the mechanism or what we know about how, on a biological and molecular level, BBSOAS develops. So I'm going to go through some of these. So Dr. Schaff this morning had gone through some basics about what we know about NR2F1. I had made these slides already, so I will quickly go through them. Let's see. I'll put all this at once. NR2F1 is a gene on chromosome number 5. Chromosomes are made up of DNA, which encode for RNA. So that process is called transcription. And so once you have that RNA codes is a template for proteins. That is called translation. So you are going from DNA to RNA to protein. And so each protein, there are tens or 20,000 or so different proteins in humans. Each protein has its own different function. So NR2F1 has its own function. NR2F1, the gene in DNA, ultimately codes for the protein, the NR2F1 protein. So just to go into a bit more detail, so I believe Dr. Schaff this morning described NR2F1 as a transcription factor. That is the NR2F1 protein, that's its role. So what that means is that it is able to control other genes. So though NR2F1 itself is a gene, by mutating just the NR2F1 gene, you can affect hundreds of other genes that NR2F1 regulates. So it essentially acts as an on-off switch for an estimated, at this time, 800 or so different genes. So yes, it can, the purpose of NR2F1 is to turn on and off hundreds of other genes in the cell. The NR2F1 protein itself can be activated by a molecule called a ligand. So I have a figure of this here. So this blue and purple blob up on top is the NR2F1 protein. A ligand, which is the orange molecule, binds to it. And so that protein then binds to DNA, and that can either turn on or off the genes that are close by. So in BBSOAS, Dr. Schaff had gone through a bit. He had broken this into two different groups. So there are deletions where the NR2F1 gene is deleted or partially deleted. And so on the top right here, or at, the very bottom right, we are symbolizing less protein by it not being there. If we move up to the top, so you could have mutations or point mutations in different parts of the NR2F1 gene, and that affects its function. So if you have a mutation in the DNA binding domain, for example, which is this bottom blue part, the function of the protein, its ability to bind to DNA and turn on and off a gene will be impaired. Alternatively, if you have a mutation in the ligand binding domain, which is this purple part, the ligand which binds and activates it may not do that as well or may not do that at all. So that will also impair its ability to be activated, bind DNA, and then affect those downstream genes. So in all scenarios, the same gene is mutated, but the exact location and the type of mutation is what matters. And so one small change in the gene can have either very little or very large effects, depending on where it is and the type of mutation. And so this can account for why just a mutation in the same gene in NR2F1 can have such variable effects. So this can explain why there's such a large phenotype, such a broad phenotype, such a broad spectrum of patients. So what does this mean in reality? So we know, we've already noted that the NR2F1 controls these downstream genes that affect the optic nerve development. So that is well defined. Recently, there is what we know about developing cells in the cortex. So the rate at which the cells divide and multiply is altered. The rate at which they develop and mature into more complex neurons is also altered. And so this has implications for the types of connections that the cells make, the number of connections to other cells, the the types of connections, where those connections are, and how they form different pathways in the brain. So Dr. Shah, by pathways I mean like the ventral stream and the dorsal stream, which is something that Dr. Shah and Dr. Edmond had talked about earlier today. And so this can all impact how sensory information is processed. So that is, in a nutshell, what we know mechanistically about BBSOAS. And so let's see. The second part is models. So this is broken into 6 different parts, and I promise not all of them will be as long as the first part. So what we know about models. Models are important because generally what we could do with cells is a lot different than what we could do with humans for some obvious reasons. For some less obvious reasons, so we can manipulate genes in cells. We could look at those cells more closely and study them more closely than what we can do in humans. And so we have a variety of different cell models. So some of the questions that we ask are there models that actually replicate what we see in BBSOAS patients? And so we do. We do have cell models that have the same symptoms, have the same phenotypes. And we also have human iPSC models, which are important because that is pretty much the closest model you could get to studying BBSOAS without actually studying an actual patient. And again, you could do things to these cells that you can't do to an actual patient. And so I put this here just to show that we are actually have a handle on all the cells that we have as far as what the mutations are, who has them, where they're being stored, what type of mutation, who has published on them, and so forth. So in terms of models, NR2F1 Foundation is in pretty good standing in terms of cell models and also in terms of mouse models or animal models. The figure that I had shown earlier, the big yellow and red grid, was from this recent review from February. They had described 112 known variants in BBSOAS in 84 patients that were fully described. And so that, since that was published in February, just a couple months later, that has gone up. So they actually have a database that's available, freely available online. And so they've gone up from 112 to 117 variants and 117 variants and 86 patients. Of these 117 variants, 93 of them are unique, which means that nearly 80% of BBSOAS patients have a mutation that no other patient, no other person has. And so this is also a contributing factor as to why there is such a wide spectrum from patient to patient. And so this is from a couple of years ago. Oops. This is from a couple of years ago. This is just a schematic showing the basic parts of the NR2F1 protein. And you might be able to appreciate that the DBD, or the DNA binding domain, is sort of a mutational hotspot. I believe that this small part takes up like 14% of the protein, but about half the mutations that occur in BBSOAS occur in this small part. So we can sort of, though each mutation tends to be unique and have its own differential effects, we can break these into general groups, and it's already well known that mutations in the DNA binding domain tend to have more severe symptoms than, say, patients that have a full deletion. So we could break generally patients into groups, and we could break animal models into groups. I'm going to skip this. So we could break, these are different iPSCs that we have, that we know of. we could break these into different groups in terms of whether they are deletions or have a mutation in the DNA binding domain, the ligand binding domain, and so forth. So we are on top of this. In terms of animal models, we can break this down into about 3 main models. So there's the haploinsufficiency model, which is another way of saying that there is a deletion of the NR2F1 gene. And so this mouse was described a few years ago by Michelle Studer's group. And so it represents patients that have this NR2F1 mutation. The model itself replicates many of the features that BBSOAS patients have. There are 2 newer models that Christian Schaff had mentioned this morning. There is the LBD mouse model, which is the ligand binding domain. So there is a point mutation in the ligand binding domain in this mouse model, and also the DBD, or the DNA binding domain. But none of these models have been published or characterized yet. And there is also a 4th model that is being planned and will soon be developed, thanks to a small grant that we had applied for back in December and were awarded. So that is in the works with Jackson Labs. Okay. Part 3, in terms of clinical resources, again, I won't go through all these questions, but there are some questions that we know the answer to. So is this disease progressive? And there's no indication of that. When is the gene expressed? That will help us determine whether or not this is a developmental disease, a developmental disorder. It will also advise us as to, in terms of treatment, when would be best to start treatment. And then in terms of how variable symptoms are, we've already discussed that. And so what is known is largely, or we can basically categorize different phenotypes, different symptoms from interviewing patient families. So this is broken down, I don't know if you could clearly see, but this is broken out into like neurological, motor symptoms, and visual or cognition. And so we get this from interviewing patient families. This is a draft of a disease concept model, which Dr. Bichelle will talk a little bit about in her presentation. She will also talk about the importance of natural history. Natural history is important for many reasons, which she'll get into, but in terms of knowing how effective a drug may be, in terms of knowing how effective a certain treatment will be, or other factors. So this is currently a part that is missing, though we are starting a patient registry today. And so that is something we are addressing. In terms of treatment resources, I'm going to go through this pretty quickly. There's only a few things that we know in terms of there are 15 or so different research groups studying BBSOAS. In terms of, is there a consensus for a diagnosis? Generally there is. So we know what to look for before we recommend genetic testing to identify BBSOAS patients. But there are other things like ICD-10 codes and certain statistics about hospitalization rates. So if the importance of an ICD-10 code would allow you to measure pretty easily why BBSOAS patients end up in a hospital, for what reason, and to be able to track that data. You will also be able to note what other symptoms they may have. Also touched a bit on standard of care. So is there a known way to what is the best way to treat a patient with BBSOAS, and then how do we standardize that to make sure that all of these patients get that same best level of care. So the fifth part is biomarkers. So biomarkers are very important for two reasons. So by biomarkers, I mean a biological marker, a way of us to know without doing genetic testing if a patient has BBSOAS. So this is not yet possible, but what would be part of what makes it important is that you would be able to diagnose BBSOAS without doing genetic testing, and then if you had a specific model for EEG, if the EEG pattern looked unique for BBSOAS specifically and not another disorder, you would just be able to do the EEG. Alternatively, there might be a blood or body fluid biomarker. So you may be able to do a simple test as the presence of a certain protein just in blood or urine. So that does not exist yet. However, it's most important because it would be able to tell you sort of down the line in a time where a treatment might be available if the treatment is effective or not. So this is pretty important. Okay. In the future, in terms of clinical trials or a future treatment, if you want to see if a patient is getting better, it will be a lot easier to see a certain amount of a protein, something that is quantifiable, and something that will likely you'd be able to see sooner than a parent would notice in a patient or their own child in terms of behavior or skills. Something that would appear in the blood that's quantifiable might be able to show you that a treatment is working before a parent would notice it themselves. And so lastly, there are outcome measures. By outcome measures, I mean is how do we quantify any of these things? How do we quantify cognitive or cognition or communication skills or motor skills? So why this is important is also because of potential treatments. So if a treatment comes along, we want to know if a patient is getting better. We want to know if that treatment is effective. And we want to be able to objectively and definitively say so. So it's important for us to have standardized outcome measures, so tests that we could give that are objective and repeatable. I'm going to skip over neurostorms for the sake of time, but neurostorms are a symptom that we believe may be part of BBSOAS, though I didn't know about it until a couple of months ago. It is not in the literature, but Melissa had told me about this, that there are these specific events that seem seizure-like. And so it's important for us to know about these neurostorms so we can measure them. And then if we can measure them, we are able to tell if they go away with treatment. So a lot of this is stuff that we don't know about yet, but I want to focus on some key steps that the NR2F1 Foundation has been doing well, which is parent and caretaker interviews and collecting natural history data, engaging with patient families and collecting bio samples, contributing to our biorepository. Currently, so in the works, I am drafting a formal research plan. In February of this year, As I mentioned, we were awarded a small grant through the Orphan Disease Center, which will allow us to generate a second or a fourth mouse model. This was done with the support of Dr. Edmund Schaff and Studer. And as sort of our grand finale, I want to introduce what we're calling the NR2F1 Collaborative Research Group. So we recently started a collaboration with Dr. Schaff, Dr. Shah, Michelle Studer, Julio Aguirre-Gazzo from Albert Einstein, and Terry Jo Bichelle from COMBINEDBrain. So we have started this research group on a promising new lead. So I had discovered a promising lead while going through the literature that we are now pursuing. So we are currently planning experiments for those and currently writing a grant to fund that. So I don't have specific recommendations, but typically we give these 4 general recommendations to our groups in terms of how do we fill in these gaps, how do we pursue research and progress research in a quick way. And so there's collaboration, which you can do in multiple ways. You could focus on data sharing and validating outcome measures. So Dr. Shah had mentioned earlier that measuring CVI is it's not standardized, it is not validated. So pursuing efforts to do that. Pursuing seed funding, so funding data collection, funding a final conceptual model, a disease concept draft that Dr. Bichelle will talk about as well. Promoting educational opportunities and also promoting access to patients. Okay, so thank you. Thank you to Carly, thank you to Melissa, thank you to the foundation for inviting me to speak. I want to also thank the COMBINEDBrain team, this new research collaborative group, and all the BBSOAS researchers. And that's it.
Okay, thank you so much, Dr. Horning. Amazing what goes on in that brain. Now you can see why we hired a scientific officer, and you can see all the fundraising efforts that you've all contributed, work on Facebook or with your families to donate to the foundation. This is a prime example of where that those funds are going to. He's the only paid person, I guess you could call, on our team. So probably one of the best investments we've ever made or continue to make. So thank you. For the sake of time, I think we will, Brian, do we have any pressing questions, do you think, from chat? No? Okay. Anybody in the audience have a question they would like to ask? It's okay if you do. Okay. All right. Well, we'll move forward. And any questions that, again, save those questions and those thoughts. Feel free to reach out and send them out to us, and we'll get back to you. We will move on. Again, thank you, Kyle. Big round of applause.